Do subscribe to Ekeeda channel and press bell icon to get updates about latest engineering, HSC and IIT JEE main and advanced videos.
Hello students, today we are studying chapter Gene, its nature, expression and regulation. In that we are studying topic Lac Operon. When I say Lac Operon, it means lactose operon present in E. coli. Let's study about this today. Lac Operon. When I say Lac Operon, it means lactose operon that is present in E. coli. Now what is this lactose? Lactose is a form of sugar. We all are aware of glucose, sucrose. Now lactose is also a form of sugar and it is used as a food source by E. coli. What do we mean by when I say operon? Now operon, I can say in simple language, is a mechanism. So what is this Lac Operon? Whenever E. coli uses lactose or lactose is present in the media of E. coli, that time lactose operon is working. So whether lactose has to be used or lactose requirement is not present is decided by this operon or Lac Operon. How does that function? Inside E. coli, the lactose operon mechanism basically have an operator gene, a promoter gene and LAC-Z, LAC-X and LAC-Y gene. Now what is this promoter gene? We all know that if there is lactose present in the media in which E. coli is residing, that lactose has to be converted into simple sugars, which is glucose, so that E. coli should use it. And to convert it into simple sugars, there has to be certain enzymes that has to be produced. Now, these enzymes will only be produced if RNA polymerase comes and binds to the promoter region. Remember the process of transcription. In transcription, mRNA or promoter region will have the information, and then once RNA polymerase comes and binds to the promoter region, that protein or you can say that enzyme will be synthesized which will break down lactose. So students, in case if the media is not having lactose or the E. coli doesn't have the requirement for lactose, don't you all think so that this binding of RNA polymerase to the promoter and then forming that enzyme would be waste of energy or waste of ATP? Obviously. In such cases, what happens is to the repressor region, or you can say to the operator segment, a repressor protein comes and binds, and this repressor proteins avoids the binding of RNA polymerase 2 to the promoter region. There were 3 genes which are involved in this segment, and the enzymes are beta-galactosidase and lactose permease. Lac Operon. When I talk about Lac Operon, what I'm trying to speak about is lactose operon that is present in E. coli, or the machinery that's present in E. coli so that it could use its sugar lactose as a food source. Now lactose operon, or you can say Lac Operon, is an example of inducible control. When I say inducible control, it means a control, or you can say a machinery which can be on and off. Basically, switch on and switch on is possible. You can switch on it whenever required and switch off it whenever required. Now, Lac Operon is basically containing three structural genes, which is LAC-I, LAC-Z and LAC-Y, which encodes for the enzymes that are required by E. coli bacterium for utilization of lactose as a food source. The three enzymes that are formed will be beta-galactosidase, lactopermeases. Now, beta-galactosidase are nothing but that help to convert lactose into usable form of sugars, which is glucose, and lactose permeases basically allow lactose to gain entry inside bacterial cell, and then conversion can be done. Now, in an absence of a medium in which lactose is absent, imagine that condition. Obviously, in that medium, the Lac repressor, or you can say a protein that stops this process of protein synthesis, binds to the operator region present on the Lac Operon, and hence no enzyme is made. Once no enzyme is made, no ATP is wasted. Students, this is an example, or you can say this is a diagrammatic example of Lac Operon. Now, what does it consist of? It consists of a promoter region. It also consists of an operator region and the three different genes, the LAC-Z, LAC-Y and the LAC-A or I region. Now, how does the process of synthesizing beta-galactosidase as well as Lac permeases start? Whenever there is lactose present in media, that time your RNA polymerase, which is related to transcription and translation, comes and binds to the promoter region. And we all know that whenever RNA polymerase comes and binds to the promoter region, at that time the process of transcription takes place. That is, the process to read messages takes place. And further, the proteins, which is beta-galactosidase and lactose primase, will be synthesized. Now imagine a condition in which lactose is not present. At that time, to the operator region, a repressor protein comes and binds. Now once a repressor protein comes and binds, repressor proteins basically block the RNA polymerase from binding to the promoter region, since they shade the operator as well as the promoter region, and hence no proteins could be synthesized. So students, whenever the E. coli is not using lactose as its food source and using something else as its food source, there is no point that this machinery or this mechanism needs to work. So hence the repressor proteins avoid the wastage of energy and prevent the formation of the enzymes that are required for protein synthesis. I hope you all are clear about this concept of lactose operon or Lac Operon. Thank you.